All right, hello everybody. I'm Professor Chuck. Some of you have probably listened to some of my audio files of my lectures, so now you can actually put a face with a name. I get the opportunity to kind of lead you through Module 4, which is our first of the two cell biology sections. And you may be asking yourself, well, okay, or you may have just been happy to recover from chemistry, so you haven't really looked into it, but you may be wondering why it takes two modules to cover the cell biology piece. This is actually one of the most important sections that you get to deal with. We've kind of laid the groundwork, and you'll hopefully see it a few times today. We've laid the groundwork in terms of trying to understand this through the chemistry sections, where it's kind of like many of you in math who say, when am I ever going to use this again? Why do I need to know this? And I'm sure many of you asked yourself the same questions. You completed modules 2 and 3 on chemistry. Really? How much of this do we really need to know? Why are they putting me through this? I hate this stuff. It's kind of like doing math. It actually had its purpose, and we'll kind of hopefully reflect that here as we go along. Also, with these next two units, they're probably, I would say, the two most important units that you understand in as much detail as you can. Not necessarily for the exam, because the learning objectives kind of lead you through that. But the important piece of this is that when we look at the body's function, basically it functions at the cellular level. Because if you think back to your levels of organization, that atoms and molecules make cells, and cells make tissues, and tissues make organs, and so on, that basically, and we'll describe that, our main functional unit of our body is the cell. And we have trillions of cells in our body, but we only have about 200 different types. And later on, when we talk about tissues, we'll describe those different types that we have. And so as we look today and going forward with future lectures in the next two units, you have to reflect that we're going to talk about basically just a general cell of the body, understanding that a muscle cell is going to be different than a nerve cell, is going to be different than a liver cell and a pancreatic cell and a lung cell, and so on and so forth. They're going to be different in terms of their structure, therefore be different in terms of their function. And so as we show cell models and we show different parts, the insides and the outsides of the cell, just kind of keep that in the back of your head, that we're describing the general parts of a human cell. And then later on we'll describe, okay, this is why this cell kind of uses some of those parts but not all of those parts. And we'll describe that here as we go along. Also later on, many of you may or may not know, one of the more difficult classes you get the opportunity to participate in is the pathophysiology class. Where we describe all of the stuff you've been going over now and apply that to how diseases work. And it becomes important for us to understand. Also, what we want to kind of reflect on for a minute is just stating the fact that with diseases, we affect the tissue at the cellular level. It just depends on how many tissues we affect. If somebody has a problem with a couple of their lung cells, so to speak, they're not going to have symptoms of lung disease. But the more and more cells that they affect with that disease, then they're going to start to have signs and symptoms and problems, and then it causes problems with other organ systems and so on and so forth. And so when we talk about diseases, and you will, and we will through this process of even this course, we're affecting the body at the cellular level, and so it's very important that we understand this. If the cell works right, we work right. As soon as the cell starts to fail, we fail too. And so that's kind of what we want to get into. So of all the chapters, and this one's kind of a big, something like 140 slides, somewhere in that range. And so, this is a big unit. And it's got a big chunks of information you want to make sure you understand, because it'll beat you up later. It's kind of using math as our example. If you miss the whole section on adding numbers, that becomes a problem forever, okay, in terms of the other concepts. So the same thing is true here. The stuff we beat you up with, the stuff I'm going to beat you up with today, is like the first and second chapters I beat you up with when you get into the pathophysiology course. So you want to make sure you reflect on that. So our cell biology, we're going to talk about parts of the cells and their functions. Today, most of our discussion will just be introducing the cell, number one, and then talking about kind of how we move things around, both in the cell as well as out of the cell. So if we look at the cell, and I reflected the fact that it's our basic unit, okay, of living things, that if you look at, let's say you look at your biceps muscle. Your biceps muscle is made up of thousands of muscle cells, each one being built in a similar fashion to all contribute to the same function. And so then again, we have nerve cells that contribute to a certain function. We have skin cells that have a particular function, but that's the basic unit that we want to discuss going forward. Now, we can actually separate different cells in our environment into two main groups. What we refer to, and if you've taken a microbiology class, you've heard these terms, a eukaryotic cell versus a prokaryotic cell. Now, our discussion in this unit will be on eukaryotic cells. And basically the difference between those having a nucleus, specifically a membrane-bound nucleus, and those are our human cells, our human cell and animal cells. When we're talking about the prokaryotic cells, kind of a big group that we deal with in that regard are the different types of bacteria. And so when we get into Module 6, we'll discuss that in more detail. But when we're talking about the eukaryotic cells, or those which do have membrane-bound nucleus, and we'll describe that membrane, and membrane-bound organelles, which are the small functional pieces within your cell. That's what distinguishes those two. Bacteria have nuclear material, but they're not contained within a membrane. And that's also what facilitates their continual division. Even while they're doing their cellular stuff, they're dividing, which is different for our cells. We have a specific time in a cell's life that it's actually dividing. We have a specific time in a cell's life when it's doing its cellular thing. So we'll describe that a little bit more here as we go along. So when we talk about our eukaryotic cells, and again, anytime you see one of these graphics, if you've ever taken a general biology class, you've seen this graphic over and over, and when you take general micro, you'll see it again, and when you take nutrition, you'll probably see it again, and we kind of beat you up with this through your academic career if science is anywhere in your study field. But we look at our three main parts. Number one, we have the cell membrane or the plasma membrane. Plasma implying its liquidity in nature, and that is true, and we'll talk about it. The molecules within that membrane that surround the cell are constantly in motion. And that's where we kind of draw the line between intracellular and extracellular, so things that are in and things that are out. As we kind of dissect, and probably muscle is one of our best examples, you have big, large muscle groups that contain big, large muscles, which contain groups of muscle cells, which then contain, so there's a level of hierarchy in terms of how we piece some of these things together. And so where does kind of one structure start and one structure end? Well, the cell membrane is the division between, okay, this is the cell, and this is the inside, and this is the outside. So we kind of keep track of where that membrane is. So our cell membrane, or also referred to as our plasma membrane, the cytoplasm is everything inside of that cell except for the nucleus. Most of that being, or a large portion of that being, actually the liquid component of the cell that we're going to call the cytosol here going forward, probably when I sit down and lecture to you again. So we have the cytosol, or the liquid portion, and then we have all the little functional pieces and parts that make that up. So if you kind of look in this graphic that I'm demonstrating here, you have this little part that looks kind of like a kidney bean, and you have these little parts that kind of look like coral, and you have these little parts that look like sloppy pancakes, and then here's the big nucleus in the middle. So all these little parts, each having a particular function, and if it functions correctly, then you function correctly. If it doesn't, then you don't live very long, or at least that tissue doesn't live very long, which can present some problems for you. So number three being the nucleus, this is where your genetics are held. So we basically have this sequestered genetic information that kind of like, I don't know, what would it be, kind of the source code, I guess, of the master code of a specific computer program, where, okay, we're going to hold this over here, nobody can touch it, nobody can manipulate it, okay, this has to stay, okay, as is, and then as that message or information goes out, then we can kind of play with it a little bit. Now, I worked in a laboratory where what many of you are going to have to deal with, or you may have already dealt with in terms of your employment, is what you refer to as your policy and procedure manual. So in your policy and procedure manual, okay, here is everything you need to basically function from the standpoint of your job, from the standpoint of how you dress, from the standpoint of for me, every laboratory test you do and how you do it, and all those parameters are all contained within this book. Well, for that laboratory, we had one master copy that couldn't be copied and couldn't be edited except by those who were deemed, authorities to be able to edit it. And so it was the master copy that didn't leave a certain place, and then kind of copy, what I want to say, so the information was disseminated from that. I don't want to get too complex into that. But anyway, that's basically how your nucleus works. It contains your genetic information. We don't want to lose that. because that's the information that helps that cell, number one, reproduce itself, number two, be able to function appropriately, and we'll describe that a little bit more. I mean, genetic diseases is where we get into that problem. All right, so let's focus on the plasma membrane first. The functions of the plasma membrane, we also called it the cell membrane, covers and protects the cell, controls what goes in and what comes out. So basically, to kind of summarize that a little bit, you need two things. You need the appropriate intracellular environment and you need the appropriate extracellular environment for your cells to function and do the things they're instructed to do by your genetics. You need a certain environment inside, you need a certain environment outside, and it controls that environment. It also controls how much water's in your cell versus how much water's out of your cell. It even controls it to that degree. We get into it later when we talk about especially the urinary unit and control of water. 2/3 of your water is intracellular, 1/3 is extracellular. You may be, why is that important? Well, it's very important. You think about dehydration on one end of the spectrum versus edema on the other end. Those become very important, and it's the manipulation of that water, both intracellular and extracellular. The cell membrane also links to other cells, so it's going to facilitate our formation of tissues. It also tells other cells who it is, and we use that both diagnostically, we use it immunologically from an immune standpoint, where, it would be great if someone needed a new tissue, if you could just give them yours without all this, problems with blood transfusions and interactions there, problems with typing tissue to say, okay, just give me your kidney, and, who cares whose it is? And it's kind of important because maybe the way to say it is, if you don't know what foreign looks like, or if you don't know what you look like, you don't know what foreign is. In other words, how does your body know that you're different than a bacterium? Recently in the news you hear about this really bad strain of E. coli kind of going through Europe. How does your body know what to fight if it doesn't know what you look like to know, okay, yeah, this is different than me, therefore. And so we do. On the surface of our cells, we'll describe them. They're actually these little purple molecules that we're seeing here contribute a large portion of that, but it basically flies a flag to say, okay, this is where I'm from. Okay, now where are you from? And we also use it, for example, if we want to target or look at cancer cells. How do we distinguish whether or not this is a cancer cell versus this one? Or what type of cancer it is. And we use those cellular flags to do that. Okay, if we look at the cytoplasm, I already described the fact that it's all the cellular soup, okay, that's inside the cell membrane. So anything between basically the nucleus and the nuclear membrane, there is one there, that's what made us a eukaryotic cell. And our cell membrane, that's the cytoplasm. So it's the cellular portion we're going to call the cytosol, okay, and all its little pieces and parts. And that'll be a big part of our discussion later. Okay, so our nucleus. This is where your genetic information is. And I like what Dr. Hutchins has done here, where he writes it very well to say, okay, this is our library. And we got different cookbooks that say, okay, this is how you make a new cell, this is how you make proteins. So it has all of our recipes. And we just hope the recipe book is put together correctly. Maybe a good example of that is, let's say you really like oatmeal chocolate chip cookies, and you have a recipe that somebody gave you and you want to try it. And they just say, just make it how I wrote it down. Well, what if they made a mistake? What if they said, add 1 cup of salt instead of 1 teaspoon of salt? Obviously, your cookies aren't going to taste very good. But if you were just instructed to make it as the recipe dictated, now you've got a problem because you're going to add 1 cup of salt because that's what the recipe says. That's exactly how our genetic diseases come into play. This is a cookbook, and the cell makes it as the cookbook is written. It doesn't interpret whether or not the cookbook is right. So if it says make this protein this way, and it does, but okay, it's a dysfunctional way. Well, it's not going to work, but that's the only way it knows how to make it. And so a cookbook analogy is a great example of, okay, this is how we're going to divide this cell and make new cells. Okay, this is how we're going to make proteins, which are going to basically facilitate the function of the cell, which is what's going to take place.